In this video, I will be going over how the cell cycle is controlled and how loss of this control can lead to cancer. The cell cycle has 4 phases: the G1 phase, S phase, G2 phase, and M phase. These phases are highly regulated by the cell cycle control system and internal control or checkpoints. The cell cycle control system is a cyclically operating set of molecules that coordinate the key events in the cell cycle. The molecules involved include cyclins and decyclin-dependent kinases, or CDKs. Cyclins are proteins that cyclically fluctuate in concentration. CDKs are protein kinases, which are enzymes that activate or inactivate other proteins by phosphorylating them or adding a phosphate group. CDKs combine with cyclins in order to activate. The activity of CDK depends on the concentration of the cyclin associated with it. Therefore, the peaks of the CDK activity correspond to these peaks in the concentration of cyclins. 3 important checkpoints in the cell cycle can be found at the G1, G2, and M phases. At these checkpoints, the cell cycle is regulated by stop and go signals from both internal and external signals. Internal signals involve signals that report whether or not important cell processes have been completed correctly and if the cell cycle should proceed or not. External signals involve both physical and chemical factors that come from outside the cell. External physical factors include density-dependent inhibition and anchorage dependence. Density-dependent inhibition occurs when cells stop dividing once the cells fill up a given space and form a single layer. Cells exhibit anchorage dependence when the cell needs to be attached to a substratum or surface to divide. Substratums include the extracellular matrix of a tissue or the inside of a culture flask. External chemical factors include growth factors, which are proteins released by cells that stimulate other cells to divide. To give an example, let's move to the G1 phase of the cell cycle. The G1 phase is where cell growth occurs. During the G1 phase, the cell must pass through the G1 checkpoint in order to continue on the cell cycle. Cells that receive the go-ahead signal and pass the checkpoint usually complete the rest of the phases in the cell cycle and divide. Cells that do not receive a signal at this checkpoint might leave the cycle and go into the G0 phase, which is a non-dividing state. The go-ahead signal could be growth factors, which would be a chemical factor as mentioned before. One example of a growth factor would be the platelet-derived growth factor, or PDGF. PDGF is used to divide fibroblasts, which are a type of connective tissue. When PDGF molecules bind to PDGF receptors on the membrane, the signal transduction pathway is activated, which allows the cell to pass the G1 checkpoint and divide. Platelets, which are a small colorless disc-shaped cell fragment without a nucleus that are involved in clotting, release PDGF when an injury occurs. The addition of fibroblasts from the division of cells helps heal the wound. After the G1 phase, the cell enters the S phase, where DNA replication occurs. During the late S phase, the synthesis of cyclin begins. The cyclins are able to accumulate because they are protected from degradation during this stage. During the G2 phase, cyclin combines with CDK, which produces MPF, or maturation promoting factor, or M-phase promoting factor. Once enough MPF is activated, the cell passes the G2 phase and begins mitosis, where MPF activates other kinases and phosphorylates proteins that promotes the fragmentation of the nuclear envelope during prometaphase of mitosis. MPF switches itself off during anaphase by destroying the cyclin attached to it. The CDK part stays in the cell in an inactive state until more cyclin are produced. During the M phase, there is an M phase checkpoint where an internal signal occurs. The internal signal is the attachment of all chromosomes at the metaphase plate to a spindle by their kinetochores, which is located in the centromere region. This signal must occur for anaphase to occur, which is the separation of the sister chromatids. The attachment of the chromosomes to the spindle activates the regulatory protein complex that activates the enzyme separase, which cleaves the cohesins that hold the sister chromatids together. Disruptions to the cell cycle system leads to cancer. One disruption would be the gene mutation in cell cycle regulators that cause positive cell cycle regulators such as growth factor receptors or cyclins to be overactive. These are called oncogenes, and the ones that are not mutated yet are called proto-oncogenes. Growth factors sending signals without growth factors, and cyclins being present in the cell at abnormally high concentrations causes cancer cells to divide endlessly and create a massive abnormal cells called a tumor. Mutations also affect negative regulators or tumor suppressors, which stop the cell in the cell cycle by making them less active or even non-functional. One example of a tumor suppressor mutation would be non-functional tumor protein P53, which is supposed to bind damaged DNA and stop the cell cycle. Normal P53 is able to bind to damaged DNA, which stops the cell cycle at the G1 checkpoint and activate DNA repair enzymes. If the enzymes cannot fix the DNA, then apoptosis occurs, which is programmed cell death. However, a non-functional P53 tumor protein cannot bind to damaged DNA, so the cell continues on with the damaged DNA. Over generations, the mutations accumulate from the damaged DNA, which could turn proto-oncogenes to oncogenes or inactivate other tumor suppressors and create more cancer cells. Another disruption causing cancer would be cells ignoring external signals such as the density-dependent inhibition as mentioned earlier. If the cell ignores these external signals, then the cell will keep dividing and stack up on top of each other. This is different from normal cells where after the cells form a single layer, they stop dividing. This is my explanation of how the cell cycle is controlled and how loss of this control leads to cancer. Thanks for watching my video.